in joining us across the country. We have got around more than 250 plus registrations. We thank you to all the participants for attending this 1 hour webinar on biomanufacturing of enzymes. So on behalf of BIRAC, I extend a very warm welcome to all of you under the 19th webinar on enzymes, biomanufacturing of enzymes under the DBT Biomanufacturing webinar series. As we move forward, I encourage all the participants to actively engage your questions and perspectives, which are vital in shaping meaningful discussions for today. And through this series, DBT and BIRAC aim to create a collaborative platform where academia, startups, and industry can exchange knowledge, identify translational gaps, and explore partnership opportunities to strengthen India's biomanufacturing ecosystem. I would also like to thank our officials from DBT, Dr. Anamika ma'am, Dr. Vaishali ma'am, and Dr. Amit Yadav from DBT to join us. Thank you so much. And further, this fortnightly webinar series BIRAC and DBT has been organizing every month. So we'd like to ask all our participants to stay tuned for all the upcoming webinars in the BIRAC website. So without further ado, I'd like to request our GGM and head in charge of technical division, Dr. Amita ma'am, Dr. Amita Joshi, to start with her opening remarks. Our MD BIRAC, Dr. Jitendra Kumar, he couldn't be with us today, so Sarah sent his regards to and his best wishes for the webinar. So I'd like to request ma'am to please give her opening remarks and welcome address. Over to you, ma'am.
Thank you, Sujit. Good afternoon, everyone. It's my pleasure today to welcome all the distinguished speakers, industry leaders and researchers, innovators and participants to this webinar on biomanufacturing of enzymes and under the biomanufacturing initiative of DBT BIRAC. Enzymes today are playing an increasingly significant role across all the sectors, including the healthcare, food, nutrition, agriculture and industrial biotechnology. As India advances towards becoming a global force and a bioeconomy leader, strengthening the indigenous processes capabilities of enzyme biomanufacturing will be a crucial step for enabling innovation, reducing the import dependency, and supporting sustainable industrial growth. This webinar would provide an excellent platform for meaningful discussions on emerging technology, the scale-up challenges and commercial pathways, as well as what are the collaborations which can take place and the opportunities which are already existing in the ecosystem and those which can be created. I would like to thank the experts, Dr. Prashant, Director, Agarkar Research Institute, Pune, and Dr. Raghavendra, Chairman and Managing Director, High-Tech Biosciences, and all the participants from DBT and BIRAC and other areas for joining us today and contributing to this important dialogue. I hope the deliberations will lead to a valuable insight and foster stronger partnership amongst academia, industry, startups, and policymakers. Wishing this webinar best of luck and great success, and wishing a very fruitful discussion ahead. I would also like to introduce our internal biomanufacturing of enzyme expert, Dr. Hemlata, and she would like to first introduce and take you to the topic before we hear our experts speak to us on what their experiences are. Thank you so much.
Thank you, ma'am. Thank you, ma'am, for your deep insights. Request Dr. Hemlata to please start with the presentation. I'll just share the slides.
Thank you, Dr. Sujit and Dr. Amita, ma'am. So, I am Dr. Hemlata Chabra from BIRAC, and today I will briefly talk on BIRAC's effort in strengthening the enzyme biomanufacturing ecosystem. Before delving into the role of BIRAC, I would like to highlight the importance of enzymes in the bioeconomy. Today, the bioeconomy is no longer defined solely by the biopharmaceutical or bioindustry. So it is expanding into much broader areas which include biofuels, enzyme solutions, digital biology platform, contract research, biologics manufacturing, agricultural biologic inputs. Collectively, these all sectors contribute to the national growth of bioeconomy. Enzymes are mainly proteins which are produced by microbes, living organisms, or the fermentation process. Enzymes are biocatalysts which makes the industrial process cleaner, energy efficient, and faster. Enzymes have application in multiple sectors, including healthcare, pharmaceuticals, agriculture, biofuels, waste management, textile industry, etc. Next slide. So the market of enzyme, it refers to a global industry which is involved in the production, development, commercialization, and application of enzymes under multiple sectors. The market size of enzyme was estimated in 2025 is 15 million, and in 2023 it is a projected market size is 25 billion with a CAGR of 6.9%. And the Asia Pacific market is one of the fastest growing enzyme market. There are a number of factors which are driving the growth of these enzyme market, which include rising the demands for sustainable and green manufacturing, expansion of food, beverages, biofuel industry, the advancement in biotechnology and enzyme engineering, the increasing application of enzymes in biopharmaceutical, pharmaceutical and healthcare industry, and the rising consumer preference for natural products. Similarly, there are major components which are restraining the growth of enzyme market, which include high production and development costs, limited stability of enzymes under the different industrial conditions, the regulatory challenges associated with the enzyme, the competition from the chemical catalysts, the intellectual property and patent issue associated with the uses of enzymes, the dependency on biotechnological advancement, and they require a high R&D and technique cost. From the Indian perspective, the role of enzymes in Indian bioeconomy, these are snippets from the IBER report 2026. The Indian bioeconomy was valued at the 195 billion in 2025 and it was 166 billion in 2024. So a 30 billion surge in 1 year. It indicates that the Indian bioeconomy sector has entered into the accelerated growth phase. So the report has identified two sectors which have contributed significantly in this year, that are the bioindustrial sector and the bioservice segment. The report has also identified four major main sector drivers. One was biofuel expansion, next one GLP-1 therapies, third is the global capability center, and fourth one is the industrial enzymes and microorganisms. So out of these four, two sectors are related to enzymes, which are underlining the importance of enzymes in the Indian bioeconomy. So this India's biofuel program, it is one of the strongest example how this biotechnology is translating into the economic and strategic gain for the India. This biofuel consumption program, which was started 2 years ago in India, the ethanol blending in petrol has reached 20% in the 2025 December, which is a key milestone in the India's ethanol blending program. So enzymes are one of the backbone of this ethanol blending program. They help in conversion of lignocellulosic biomass into fermentable sugar. Enzymes helps in production of ethanol with the agricultural waste, crop residues. The advanced enzyme cocktails are producing ethanol in a faster way, in an efficient way. So overall, the enzymes are making this bioethanol production faster, cleaner, and economically feasible. Next slide. So enzymes are one of the key enablers in the bioeconomy. So now coming to the BIRAC. BIRAC is a Section 8 not-for-profit company established under the aegis of Department of Biotechnology in the 2012 with a vision of to stimulate, foster the strategic research and innovation and strengthening the Indian biotech industry, including the startups, SMEs, industry, to create affordable products which are addressing the unmet need of the society. Next slide. The major role of BIRAC is to support early and late stage innovation research. So the BIRAC support startups, industries, SMEs from ideation stage to commercialization stage through its different schemes, including BIG, SIBRI, BIP, PCP, the other schemes. Apart from funding, it enables the different services also like IP management, technology transfer, bioincubation infrastructure, mentorship for the regulatory challenges, product commercialization through the strategic partnership. There are a different sector where BIRAC support, including device, diagnostic, drug delivery, vaccine, veterinary science, environment, agriculture, energy, and secondary agriculture. These are some of the examples of BIRAC supported projects in the area of enzymes and industrial biotechnology where technology has reached at the TRL 7 and above. So some examples are like cellulosic ethanol plant for rice straw management, development of a novel cocktail of cellulosic enzymes for deconstruction of lignocellulosic feedback, viable enzyme production using agro waste as a raw material for industrial feed and healthcare, indigenous production of dextranase, pilot scale technology facility for value-added chemicals from biomass. Next slide. Production of beta-galactosidase during agri-waste, production of thermotolerant and acid-stable phytates. So these are the some examples. Next slide. So coming to the Bio-E3 policy, the Union Cabinet chaired by the Honorable Prime Minister, it has approved Bio-E3 policy in August 24, 2024. And in the presence of Honorable Minister of State, Dr. Jitendra Singh Ji, the policy was launched on August 31st, 2024. The policy has laid down the framework for the implementation of biomanufacturing and biofoundry initiative. The policy aims for a green, clean, prosperous and self-reliant India. The policy aims to empower Indian institutions and industries to engage in transformative innovation through public-private partnership. So, under this Bio-E3 policy, the 6 thematic verticals of the national importance have been identified. So, to augment translational across these six, the translation across these 6 verticals will be augmented through support research and through establishment of bio-enablers, that is bio-artificial intelligence hubs, biofoundry, biomanufacturing hubs. So these are the 6 thematic sectors. So in the first thematic sector, bio-based chemicals and enzymes, the first call was launched in the month of April 2025. So the call focus was on to enhance the indigenous enzyme manufacturing capability of India. This call's focus was on first focus was on enzymes for the biotransformation of low-cost feedstock into high-value functional biomolecules. The biomass hydrolyzing enzymes focusing on the efficient conversion of lignocellulose biomass into fermentable sugar. The enzymes for biotransformation towards production of specialty chemicals and APIs. And the proposals were invited under 2 categories: discovery and application oriented, and the bridging the gap for scale up. So this call is closed, but I would like to request all stakeholders to go to the BIRAC and DBT website for future calls. Thank you all.
Thank you, Dr. Hemlata. I think that beautifully captured the overview of BIRAC efforts in fostering ecosystem growth in biomanufacturing of enzymes. Now I'd like to request Dr. Prashant Dhakefalkar sir, Director ARI Pune, to please enlighten us on the role of academia in fostering biomanufacturing of enzymes. We're really honored to have sir over here with us today. And I'd like to request the participants who have joined us, please put your questions on the chat box. Few of them will be taken up after the talks from our speakers. Please stay engaged, and rest we can discuss the same post-webinar as well. Thank you. Over to you, sir.
Thank you so much. Can I have the slides, please?
Yes, please, sir. Slides. Can you put the slides?
Yes. So at the outset, I want to thank DBT BIRAC for giving us this opportunity. I want to congratulate them for the excellent initiative and organizing such presentations where both academicians as well as industry personnel will be immensely benefited. I must also admit that I'm slightly in the awe of my next speaker, Dr. Raghvendra Gaikewari, who is a very astute industrialist and adroit academician who has a wonderful blend of academic flavor as well as industrial success, and would be eager to listen to many of the success stories from him. But as the title slide says, I am very happy to present the role of academia in fostering biomanufacturing of enzyme. May I have the next slide, please? So roughly it will be divided in 6 parts. And first and foremost is I would like to discuss about India's enzyme import dependency, the gap between the knowledge and marketing. This will be followed by Bio-E3 policy vision. I would not now spend more time on it as Dr. Hemlata Madam has already explained to us very nicely. So I will go ahead and directly talk about academia's contribution in enzyme ecosystem. I would also talk about the application readiness, libraries, code development, etc. The five pillars of academies that are very important for the industry and what must change. I would also discuss about the possibilities of collaboration and eventually how we want to reach our goals by 2030. May I have the next slide, please? So, roughly, I mean, before we get into the details, let us discuss why we are talking so much about enzymes. And whenever we talk about it, I always like to give a very simple example. For fertilizers and everything, we have to have nitrogen which is fixed, which can be used in the form of ammonia or any other gas. And whenever we have to do that conventionally, we use the Haber-Bosch process where we heat it in presence of a very effective catalyst at 700 to 1100 degree Celsius, and at such high temperature after 7 to 11 hours we get the ammonia which is a product of this. So what I am trying to tell you here is how energy intensive and laborious this process is. Now just think about microorganisms, the nitrogen fixing microorganisms which have nitrogenase enzyme complex can achieve the same at ambient temperature in aqueous environment with the help of their nitrogenase enzyme complex, and that too in few minutes. That is the power of microbial enzymes. That is the power of any enzymes. They are the most efficient green alternative to chemical processes, enabling sustainable manufacturing. And that is the most important advantage the microbial processes or the enzymatic processes have over the conventional chemical processes. And most importantly, the economic drive is the most important one, especially in today's scenario with the advent of CRISPR-Cas or some of the advanced molecular techniques, and also in the presence of AI tools. With the availability of such tools, I believe that biomanufacturing is going ahead in leaps and bounds, and this is something which is going to be a great time ahead for the environment sustainable, techno-economically viable production of various chemicals. And that is where the India's Bio E3 vision, which is economy, employment, and environment, and the net zero goal for sustainable technology development is something that is going to be possible. Next slide, please. So, when we talk about it, why are we discussing about Bio E3 vision so much? Well, when we talk about the enzyme or global enzyme market, as Madam has talked about $20 billion soon approaching, we already know that it has crossed 14 to $15 and it is growing at annual CAGR of approximately 7%. When we talk about it, we realize that when economy is growing at such fast rate in sector of enzyme technology, Indian share is relatively much less. Next slide, please. So I will not go into details of all the industries where enzymes are applicable, as Madam has already said, but this is a very interesting number. India's annual enzyme import bill is $200 billion. And why is it surprising? Because in India, we have various academic institutions. We have Indian Institute of Science, we have IITs, we have ICERs, we have DBT institutes, we have DST institutes, we have CSIR labs, and various industry R&D centers. We have more than 10,000 enzyme related papers and every year that are published from these institutes. They are high impact paper. There are mechanistic insight into many of these enzymes and their mechanism. But when we think about how many of these inventions or discoveries are getting translated into actual commercialized technology, it is not even 5%. When we think about the $15 billion plus world economy growing at 7% annual growth rate, then it is the $200 billion market in India, which is where we are importing. It actually is not acceptable. But what we want to see is this actually is presenting us a great opportunity, and there is an important role that academia can play in closing this particular gap. How we will see, we will discuss. May I have the next slide, please? So let us just understand when we talk about global enzyme market and when we consider or compare the Indian realities, how exactly is the scenario? The global market, there is explosive growth. There are broad applications in several fields, and it is also have established proven beyond doubt that circular economy is possible only with enzymes which are central to biomass valorization and eco-friendly processes. We have already listened to so many of the DBT or BIRAC sponsored projects where this has made possible. But what is the Indian reality today? We depend heavily. You visit any of the pharmaceutical industry, and if at all they are using lipase, you will see that it is always made in Denmark or made in Germany or made in USA. When we look at any of the other enzymes, let that be protease in detergent industry or let that be any other enzyme, almost always it is imported. Even in spite of all such steps that we have taken or DBT BIRAC has been taking, the reality today is the enzymes that we are using in saccharification of lignocellulosic biomass is always imported. So when we have such fantastic academicians, when we have such fantastic infrastructure, where are the bottlenecks? Where is the infrastructure gap? And that is something that we need to discuss and we need to indigenously breach this gap and we need to ensure self-reliance. And it is very important, especially in today's era when rupee is devaluating or depreciating against the dollar every single day. It is important that we reduce our reliance on imported goods and we start supporting Atmanirbhar Bharat mission or called by our Honorable Prime Minister and we start manufacturing almost every single chemical in India, at least all the enzymes that our industry require in India. May I have the next slide, please? So what does academia bring to the table? When we think about it, we have fantastic laboratories. We have very intelligent, well-trained manpower. And all these people are utilizing the infrastructure that is available for discovery. They are investigating the biodiversity. They are using even the metagenome or other the sophisticated state-of-the-art technique to unearth the novel catalytic functions that standard screening methods may miss. And this is something that has to be done extensively, and this is very well done by the academic institutes. They are with the help of CRISPR-Cas technology, with the help of directed evolution, and with rational design to transform wild type enzymes into hardened industrial workhorses is something which is very much possible. We just listened to the biofuel economy and we are talking about biofuel technology, and there also this CRISPR-Cas assay has been extensively used for the production of isobutanol by blocking various organic acid production pathways and directing the enzyme the metabolic flux towards production of isobutanol, thereby increasing the yield from 2 gram per liter to more than 36 gram or in some cases in latest report 61 grams per liter. And that is something which is possible because of the various techniques that have been mastered by the academician that can be very effectively used. The process development which requires the breaching of the biology and engineering. Most of the times academic institutes have been developing the processes which can be termed as proof of concept, but for the scaling up we require the infrastructure for scale up studies, and such kind of infrastructure can also be offered by the academic institute for the benefit of the industry who can practically use this data to confidently evaluate the techno-economic viability assessment. And most importantly, the academic institutes, they bring the human capital, the talent that we have, the very well trained and brilliant minds that we have in science, if in form of the graduate PhD students, MTech students, postdoc, it is unbelievable. Many of them have trained abroad and have come back to our country. And this is something that we can use, the academia can offer to the industry. May I have the next slide, please? So what academia is currently contributing is exactly that. When we talk about such kind of research, we know that many of the academic institutes all over the country have used metagenome approach as well as culture dependent approach. They have investigated the microbial diversity associated with extreme pristine and other niche habitats. They have developed the metagenome libraries. They have investigated the genomes, the genes, the three-dimensional predicted structures of the protein, getting insight into catalytic activities of these enzymes, so on and so forth. So essentially what academic institute have done very effectively is they have made the bench work, they have completed the bench work, and all these databases now available. What is important is this database must be very freely available to the industry or for the industry, and it should be used. IP generation is another part where academia has done very great, and the patent filings from IITs, ISCs, CSR laboratories, and many other DBT and DST institution has been increasing, and there are very many novel enzymes that have been reported, and this has been recognized globally every time, every single paper that has come out. And when we look at the papers in the Nature, absolutely fantastic, brilliant contribution. The only thing that we now need to do is convert that Nature paper into a scalable technology, because Nature publication will not guarantee a scalable technology, and that is something that we academicians must accept and start doing, taking some steps towards such scalability. The next slide, please. So what exactly is the gap? If we just look at this particular pipeline, it is very interesting. As I said, there are tens of thousands of publications that are coming out of academic institutes, and every single step there is approximately 50% drop. So of this, whatever are the novel enzymes that have been identified, novel in terms of their amino acid structure, novel in terms of their substrate profile, their catalytic efficiency, their temperature or solvent tolerance, their maybe enantiomer selectivity, so on and so forth. So there are several such enzymes that have been reported. How many of them are protected for intellectual property? You will see that there is more than 55% of such discoveries are not protected for intellectual property. Then when we have to optimize all these enzymes for their production, for their substrate profile and several such things, not even 22% of that is reaching the step of proof of concept or TRL3 or TRL4. And if at all they don't reach the TRL3 level, then the pilot scale is obviously out of questions. And only 10% or even less than that is reaching the pilot scale. And I'm talking about 100-liter plus fermentation demonstration. That is important to have some sense of techno-economic viability assessment, the insight into scalability of the process. And what percent are commercialized? Not even 5% are commercialized. And this is something which is very, very important, and we need to breach this gap. May I have the next slide, please? So there are 5 pillars and expectations from academia. The first and foremost is application-ready research. Publications in Nature or any high impact factor journal is always welcome, but industry is not going to be impressed with that. Industry will always ask for real world validation data. If I talk about amylase enzyme and if I say that this is fantastic enzyme, this can saccharify starch like anything, industry, and this is its Vmax, Km, kcat, turnover number, and all 3D dimensional structure and the substrate binding groove and the catalytic groove, I talk about every such thing. An industry person will ask me a very simple question. If at all I put 10 grams of starch, what is the minimum amount of enzyme that you can put to liquefy that starch completely? They are not interested in anything else. Show me the liquefaction of the starch and I am convinced about the utility of that enzyme. That is something which is very important. Most importantly, we always publish papers and we talk about so many libraries of enzyme that we have prepared. Maybe those the libraries have been prepared from various metagenome libraries that we have, or maybe even from the microbial cultures that we may have cultivated in the laboratory from that, or maybe we can gain some insights into the genomes that have been sequenced and their enzyme coding regions have been well characterized. Well, it is very important that all these libraries are made available to industry. Only then it has some importance. Otherwise, we have seen hundreds of thousands of cultures or clones or libraries that have been prepared but that have remained within the confines of the labs and have not reached laboratory. And that is something that we have to change. The co-development model is very important, and the success lies in embedded partnerships. These partnerships with the industries cannot be just used for getting some tax benefit. That has to be right from the beginning, where we understand what exactly is the requirement of the industry, what exactly is the problem that industry is facing, and what type of solution is going to be useful for the industry. So there has to be a very regular, routine, frequent meetings with the industry, sharing of the data between academia and industry, and based on that, then industry should develop the confidence. So that has to be relational, that has to be transactional. That means the technology is developed, not only remaining stagnant at the level of the proof of concept. The fastest TTM mindset is also very important, because whenever an academic institution thinks about any of the project, we talk about 5 years of timeline, because that is accepted time that a PhD student will take to complete his doctoral research. But that is definitely not something which is very useful. We must run, or we must work at the pace that the industry requires. And industry will talk only in terms of months. They cannot talk in terms of years, because by that time the ship has already sailed, and that is very important. Also, what is important for the academicians is to understand the regulatory requirement. The regulatory literacy is very important, and it must be imbibed in our mindset that my technology should fulfill all the regulatory requirement. So genetically edited microorganism is far more suitable for industrial applications or commercialization than the genetically modified organism. And whenever we talk about proof of concept or my technology, we should not talk at any scale less than 100 liters. And then only the industry will believe. So the valley of death that we call the proverbial valley of death, where many of the technologies are dying and not proceeding beyond TRL 5, that should not happen, and we will sail beyond that. May I have the next slide, please? So when we talk about lab discovery to commercial product, we must have these 5 points in our mind: the application ready research, the shared enzyme libraries, the co-development model with the industry, the TTM mindset, and regulatory and pilot scale requirements. That is regulatory literacy and demonstration at pilot scale. And only then we will be able to cross over the valley of death for almost all of our technologies, and we will be able to bridge the gap between our laboratory research and the commercializable research. Then only lab discovery will reach the commercial product. May I have the next slide, please? So there can be different models, and we should always discuss, and I always like to discuss the model of Novozyme, a Danish tech company. Now it is very interesting to remember that though this company has been immensely successful, it started all its venture in close collaboration with Danish Tech University. And only in collaboration with that university, they developed several libraries, they developed several technologies which were commercialized. That technology, that associationship or partnership was long term. It was embedded research partnerships. There were always IP agreements which were shared right from day one. Their problem definitions were joined. That means whatever problem was there, it was jointly defined by the academia industry together. Their technology transfer pipeline was accepted. There was a very reasonable profit sharing model that was developed. And today we see that Denmark has, or rather Novozymes, which is coming from a very small country of population of not even 6 million people, but it is probably the number one company in enzyme sector in the entire world. And we should derive a lot of encouragement, inspiration, and some lessons from this particular association. The next slide, please. We have also seen in our association with many of the industries that only when you have a very open type of associationship, only when you give up a typical academician's approach of transferring technology on as is where is basis, only when you own the responsibility and say that my product will work. If it doesn't work, I will put my best to make it sure that it works in your lab. And if not, then we will again take back our record. So with that kind of confidence, when you approach the industry, more often than none, your tech transfer helps. It is successful, industry is convinced, and most of the times, even though they may face some problem here and there, together you can always overcome the problem. So that is why there has to be a deep shared research agenda. One should always agree on shared IP from day one. But shared IP means IP is filed together by academia and industry. But at the same time, academia should not expect to sell the same IP to multiple industries. Industry is in the business for making profit, and they will have the inclination towards sharing IP only when they know that their IP is protected in one way or other. Also, the timelines, the long timelines should be done away with. No project with industry should exceed 12 to 18 months. There, the start point, the problem should be defined right at the beginning, and the relationship should be, the talent flow should be rotational, embedding. The internship should be incorporated in the agreement. There should be free movement of the industry personnel to the lab and from lab personnel to industry. It is very important to show confidence and faith in each other. Many a times it is believed that if the industry person comes to my lab, then he may look at some of the other projects and may get some idea and then he will develop. Well, industry would never do that. I mean, they would rather find out that you have made some progress in one area, so together they would like to progress it, because the expenditure is going to be much lesser for the industry, and that is something that we academicians should accept and move on. And our product, publications, and talent should be embedded in our partnership. May I have the next slide, please? So that is why we should have these particular lessons. For academia, for academicians, I believe that we must follow these 5 steps. First, audit our research for commercial readiness every year. At the end of year, in the month of December, one should audit that in entire year, what research did I do and how many of my research findings are ready for commercialization? What steps did I take? What did I miss? If I missed it, how I am going to overcome that in the early part of the next year. We should be committed to more than one industry defined collaboration per group. So there is no reason for us to shy away from fundamental research. Fundamental research has its own place and it is very important. Only when we are fundamentally strong, we would be in position to develop this. But only because I am involved in fundamental research, I will not be doing any industry or applied research. That kind of mindset needs to be changed. Train the students in techno-economic assessment, scale-up principles, and basics of intellectual property protection. Advocate for empowered and well-defined TTOs. Engage BIRAC funding, I can give that, or there are several other partnership tools where we can apply together for funding. At the same time, industry need to open up a bit. They need to open up and share real technical problems, not a sanitized version. Only when they have this open mindset, the academician would realize what exactly is the real problem and he would be able to give his best. Invest in relationship before breakthroughs are needed. Otherwise, it becomes a very difficult thing. If at all you try to get into a relationship after the breakthrough is there, then neither the academician is uh, willing to change. He would be more or less inclined on selling or transferring his technology on as is varies basis. Co-funded pilot scale validation is very important. Student rotation is also important because these students who have worked on the project at this stage, when they are trained, they can go to the industry and have a better insight and define success jointly, not individually. Only when we call it a joint success, then only our partnership will be very important. Let that be in the form of papers or let that be in the form of products. So this is what there are several ways in which we can compare. We can go on to the next slide, please. So this is my second last slide. So enzymes have been doing their job for 3.5 billion years with precision, efficiency, and no waste. And that is the model. And that is why when we have these enzymes, when we have the science, we have the policy framework, we have the talent, now we need to have the will and architecture to connect them. And I believe that BioE3 has lit that flame and let us carry forward together. May I have the next slide, please? This is with this slide. You can read it, and this is the message that I would like to give. And I would like to again thank BIRAC for giving me this opportunity to share my thoughts. If there are any questions at the end of the session, I would be happy to answer them. Thank you so much. God bless.
Thank you so much, sir. Thank you. I think the entire discussion beautifully highlighted how academia can drive scientific breakthroughs while supporting scalable and industrial biomanufacturing. So thank you so much for that, sir. And we'll take up few questions after the talk from our Gaikwadi, sir. So request Dr. Raghavendra Gaikwadi, sir, Chairman and MD, High-Tech Biosciences India Limited, Pune, to please join us, sir, for your thoughts on opportunities and challenges in biomanufacturing of enzymes. So a lot of industry and academia, and mainly the startups, they're looking forward to hear from you today. Thank you, sir. It's an honor for us for you to join.
Yeah.
Please.
Thank you. Thank you. Yes. Can you please upload the presentation?
Yes. We're doing that.
To start with, I just want to thank Dr. Prashant because he has really put the academia perspective so cleanly, and it has been reflecting a very important aspect that the academia in India is also changing and becoming pro industry. So that is something which is very interesting. Just to add to that, I am working with Dr. Prashant's institute for more than 6 years, and it's really a fruitful association between industry academia that we have come out with fantastic technologies with the help of academia. So role of academia cannot be really ignored when it comes to some sort of an technology development. So to start with, very important aspect of enzyme biomanufacturing. So it is beyond fermentation. I'm not going to talk anything about fermentation or industry aspect of the fermentation, but I'm going to talk with reference to a specific questions that industry should ask before entering into the enzyme biomanufacturing. So can we go to the next slide, please? So why enzymes matter now? That is very important. The first and foremost is biology is now replacing chemistry. That is happening in a very big way. Even if you consider the APIs, people are shifting from 1 gram 500 milligram dosage to 1 milligram dosage. And for that matter, you require completely an enzyme system to really work for that matter. And that is the reason. The first important aspect why the green chemistry. So because customer want lower carbon footprint, that is something which people are really looking at. Lower energy consumption, that is another aspect why people are looking at the green chemistry. Fewer solvents, that is a specialty chemical industry demand that you require fewer solvents for that matter, and better selectivity. So this is the driving enzyme adoption, that is what I feel. So next industrial revolution will be biology driven by all means. It is not going to be a chemical driven anymore. Chemistry has reached to its level of supremacy. Now, second important thing is that any technology becoming much viable, the enzyme are becoming more viable because of the sector convergence. Pharma, food, agriculture, and sustainability. All the aspects will be leading you to use the enzyme, and that's why enzymes are now mattering more. Resource efficiency and India's movement. Now, that is something which is very important. India has once in a generation chance to become a global biomanufacturing powerhouse because each and every business really look for 2 to 3 suppliers. Now China is the first preference, whether we accept or we don't accept it. But the second preferred option will be India. In this case, this is the right moment for India to encash and establish our biomanufacturing establishments. Can we go to the next slide, please? Now, what is the real industry story? Now, this is something which we have to really understand. 20 years back, it was a very simple enzyme business. Ferment, purify, and sell enzyme. Purify to 100% purity for the pharma applications and for industrial purpose, some crude enzyme preparation, and you can sell the enzyme. That was the enzyme business that was happening 20 years back. Now the scenario has been completely changed. Today, the customer is not looking for enzyme. The customer is looking for the solution. A feed manufacturer wants better feed conversion. Pharmaceutical company wants a granule synthesis route. A chemical company wants to eliminate hazardous solvents. And at the same time, ethanol producers want better conversion efficiency. So everybody is looking for some solutions and they are having their problem in place. Now the role of biology comes into a picture. Whether the biology is having an answer for these problems. And if the biology is having the solution in the form of enzyme, then people are turning to a data. And the moment they are turning into a data, AI, bioinformatics, protein modeling, and genome mining are changing the way the enzymes are discovered and optimized. After that, whether finding an enzyme is sufficient or whether we are having the process from where we can take this solution from flask to a fermenter, and from fermenter to a commercial plant, and from commercial plant to a profitable business. So these are the 3 major aspects, and every startup company really start looking at these 3 aspects, because most of the technopreneurs, they end up by finding a fantastic title and then they are very happy about it. But the problem lies in a scaling up, and from scaling up to a commercial plant, and from commercial plant, whether you are having the right business proposition for that matter. If it is not there, then technically brilliant enzyme without a business model remains a publication, or at most it can be a patent. A technically good enzyme with a strong business model can become a successful industry. This very important aspect everybody has to really look into. Can we go to the next slide, please? So what is there in the enzyme development? First is the strain selection. So if you are knowing what problem we are going to solve it, then we have to find out which strain we are going to really select, in which form we want to really express our protein or enzyme, what will be our fermentation strategy, fed batch or a continuous or a solid state, choosing the right mode of a scale, that is very important, and evaluating at each and every stage. Then followed by downstream processing. Now this is something which is very important. People are talking about a titer, because I am on many of these advisory boards of DBT, BIRAC and those type of things, and I really evaluate many proposals, and people are talking about titers, but very few proposals addresses very important aspect of a downstream processing, because in the industry 50 to 70% cost of the production is directed towards the downstream processing, and unless you are having a very robust downstream processing, you cannot have a commercially viable process. Followed by that, formulation and stabilization. Whatever enzyme you are going to have, how you are going to stabilize your enzyme, how you are going to formulate that, because If the application site is somewhere else, the transportation aspect has to be really considered for that matter. And the last but very important, regulatory and scale up. The scale up, many times people do pay attention to, but regulatory should be also looked into from day one itself. It is not something that at the time of a launching of a product, I start searching for the regulatory aspect. It is not really a good sign. It will really lead you to a disaster. Can we go to the next slide, please? So when I'm saying that 50 to 70% of your total production cost is into a downstream processing, because media cost has to be optimized. But media cost is costing not more than 6 to 8% when it comes to a fermentation. Second important aspect is oxygen and heat bottlenecks. Now at 100 ml flask, oxygen transfer is not a problem, but the moment you scale up your fermentation from 10,000 liter to 1 lakh liter fermentation, at that time oxygen is the most expensive affair to really use into a fermentation, and that is becoming a major consumable cost, even sometimes equivalent with your media cost. That's something which is very important. Then what is important? Peak yield matters far less than the batch to batch consistency. This is something which is very important in many of the R&D labs of industry or in academia. Many times the report given even by the R&D of the industry, the peak titer which has been given, and then that really that many times are disastrous. But a consistent yield for 10 batches, that should be considered while you are scaling up your processes. And one more thing I just want to add, the heat bottleneck. Now, for example, if you really look at the locations at which the China has put their biomanufacturing facilities, which is producing enzymes, is Shedong, having a temperature, average temperature ranging between 11 to 14 degrees centigrade. Then Jiangsu, where the average temperature between 14 to 16 degrees centigrade, and Jiang is another province where the average temperature is 15 to 18 degrees centigrade. Maximum is not more than 32 in any of these places. Now this is something which is very important. If your maximum temperature is going to around 42 degrees centigrade, by any chance in enzyme manufacturing, your temperature or the utility cost so as to maintain your temperature in a fermenter ranging between 0.5 paisa, half a rupee to nearly about 3 rupees a liter. So just consider around 1 lakh rupee or 1 lakh liter of fermenter, you will be paying nearly about 9 crore rupees just to maintain the temperature. And if you want to really maintain your EBITDA at 33%, you have to really go for 27 crore rupees of a higher turnover from the same facility. So that is something which many people really ignore. So when you are selecting a facility where you will be putting up your plant, at that time seriously think about what will be my utility cost. That is very important. And downstream processing, as I've told you, it is very expensive in case of protein engineering and in protein production or enzyme production. So it has to be very properly studied, and engineers should be involved in the process development from day one. Even at the laboratory level also, you have to really visualize how the scale up operations will look like, and then eventually you have to define your complete process. Can we go to the next slide, please? So what startups usually miss? Many times the startups are coming from their postdoc or some sort of a PhD work. They have completed, they have got some fantastic result in that one, but they don't many times study what market is asking for. Whether the market is asking for an enzyme or market is asking for end-to-end solution for their problem, in which one component, one tool is an enzyme. That aspect the startup has to really look into. And if market is asking for some solutions, in that case they have to really package their complete company towards not only making an enzyme but making a complete solution for the end users. Then big techno-economics is something which very important. Underestimating the cost per kg, scale up cost. Many times the what we say that many times like the enzyme cost they consider at which the market will be selling, it will be something near about 2 to 3 times more than what actual market is really buying the material is. So this big techno-economics is also very detrimental for startup. Then ignoring regulatory pathways. So you have to really be sure about your regulatories, considering whether am I been allowed to use BL21DE3 star or not. Two, how much time it is going to take a permission if I really want a 10,000 liter fermenter. So from day one, your regulatory person should be there in your team. Then flask to plant directly. Many times people don't really go for the intermediate stage where they have to spend near about 3 to 6 months to really scale it up and having all engineering constants in hand before really going for the scale up. So sometimes this type of mistake people are doing it. So take your own specific time for the scale up, do scale up and then go for the plant. And lack of application validation. Enzyme that performs in the lab may fail completely in the customer's real world industrial process. So this is something which is very important, like what is the environment in which they are going to produce. I will just give you an example. A very big company has developed some sort of a aflatoxin degrading enzyme in E. coli. Complete scale up and everything was developed. And when they really purified an enzyme, traces of endotoxin was present in that aflatoxin degrading enzyme. A complete project has to be really shaped. So these are the very important aspect people has to really look into, because your assumptions when you really scale up and produce, those assumptions should be very strict from day one. Can we go to the next slide, please? So the future is not just enzyme. Now, enzyme engineering has become much faster than it was near about 3 years back because of the AI tools which has really come into the picture. The directed evolution, the wet lab requirement of a directed evolution is no more required. You can really have in situ or in computational field, you can have the modifications of the enzymes and then have the clones of those enzymes, and getting an enzyme for a particular application can be really designed very well and much faster. Then CRISPR strain development is much faster. That is also very important. Wholesale biocatalysis. This is something which people are ignoring, but many times rather than going for enzyme purification or crude enzyme, people are preferring to go for the wholesale biocatalysis, and that is really reducing the cost of the application. So rather than just selling an enzyme or making an enzyme, can I really make a complete technology where I'll be selling an end product and enzyme will be my intermediate, which I can use for the biocatalyst, for that biotransformation. Then continuous manufacturing, whether the manufacturing should be a fed batch type or it's a plug flow type or it's a continuous manufacturing, those all aspects has to be really considered from the beginning. And personalized wellness enzymes. So that is also now picking up like anything in the field of enzymes. Microbiome linked enzyme targeting precision health, nutrition and personalized medicines are taking very important place nowadays. So from that angle, we have to look at industrial enzyme because the demands will be huge in coming years. Can we go to the next slide, please? So India has got a great opportunity. We are very strong in chemistry background. Then there is a fantastic pharma ecosystem, and 70% of our API intermediates are coming from China. So it itself is having a great opportunity. Rupee is falling. Dollar is becoming much more stronger. Import is becoming more expensive. It's important that this ecosystem is going to really be very happy for you people to really, if you are able to give them a solution with some form of enzyme or a technology. Then lower manufacturing cost in India. Still, India is still having a comparatively lower manufacturing cost. Growing biotech talent. Now that is what Prashant sir has really explained to us, that there is a really very good pool of people who will be easily available in the field of biotechnology. And global supply chain diversification creates once in a generation opportunity for India. That is what I mentioned earlier also, because supply chain always look for a diversification, and the second destination is India, and we have to really encash on that one. Can we go to the next slide, please? So the global enzyme market potential is 2023 data. Somebody is saying 13, 14. Doesn't make much of a difference. Even if I am having a 20 billion of market in the world, how much I can really cater out of it? Even if of that, around 20 million I can get it. It can be a very big biotech company. So we have to really look into how much business we can generate through this 6.8% CAGR. And second important thing is that in the field of biotechnology or in IT industry, you're seeing constructive destruction is something which is happening day by day. So suppose my aeration cost is very high, can I really think of some sort of an anaerobic system where the enzyme can be produced without really causing me much cost of the aeration or something like that one? That type of some disruptive technologies can be thought of, and we can really have the edge over others. Can we go to the next slide, please? Unfortunately, India is having 4% of the total market. 65% of the enzyme we are still importing, and India's growth market is 10%, something 10% year on year. So that is something which is very important. Now BIRAC is really investing good amount of money so as to create an infrastructure. So scale up infrastructure at present is not available. Within next 2 years time, many such infrastructures will be available. Enzyme engineering depth is not available. Many of the IT companies are working separately and the wet lab are working separately. It is not going to solve the problem. Like wet lab and IT should really come together. That AI tools should be used in a very close vicinity with the wet labs, and then we can really come out with some sort of an good enzyme platforms available. Then regulatory clarity, slow approval pathways for novel enzyme applications in food, pharma, and agriculture. That is really a serious problem in India, so it has to be addressed. And investment in biotech R&D. India spends 1% of less than 1% GDP into the R&D as against of 3%. So if you really look into the total investment, our GDP is not as big as China or US, and 3% of that GDP is going into R&D of the biotech. So just consider that we are nearly about 20 to 22 times less investment into our R&D, and that is affecting our speed of implementing our R&D into the real technologies and commercial viabilities. Can we go to the next slide, please? So a different way to think, like, can we make an enzyme? That is not the question at this moment. Which industrial pain point can biology solve better than the chemistry? That question has to be really answered very properly. So Prashant sir has said it very rightly that we have to really go and talk to industry, like what is the problem the industry is facing. At the same time, industry also has to be open with their counterparts to really tell them these are our pain points. But as a startup companies, you have to really understand what is the problem of the industry and whether your solution will able to solve their problem or not. That is very important aspect of this question, that industrial pain, am I really solving that or not? Like, for example, we worked on one phytosterol to pregnenolone development, and the titer expectation is around 20 gram per liter. But if you develop something less than 10 gram per liter, it is of no use. So this is something which very important aspect you have to really look into. So case study in mindset is API intermediates, waste valorization, animal feed enzymes, probiotic associated enzymes, and green specialty chemicals. Now this aspect is having a different problems, and you have to address the problem and not providing only enzyme for that matter. Like for example, animal feed enzyme, phytic, xalanase, and protease enzymes improving livestock nutrition. Now suppose if I'm giving my enzyme preparation for 1 rupee, and my the mass of the like if I'm giving it to the cattle, for example, and the milk rate has been increased by nearly about 10%. So if I'm giving a 1 rupee of enzyme and the 10 rupees yield has been really improved upon, then the industry will ready to buy your enzyme formulations. But for the heck of it, just I'm adding some enzyme into that one, and for 1 rupee I'm getting a benefit of 1 rupee. Nobody is going to buy my enzyme. So this aspect has to be really looked into. Can we go to the next slide, please? The future winners will not be the people who manufacture enzymes. They will be the people who integrate biology, process engineering. I am emphasizing process engineering because that is very important, and many biologists ignore that, because process engineering, which is actually controlling your downstream processing, and downstream processing is having around 70% of manufacturing cost. So it has to be properly addressed. AI and business into a scalable solution. Now, this aspect is something which is very important. Why I'm saying to you about business angle, because business is something more than commercial production of the product. Like suppose, for example, you are working on enzyme, you got a strain, scaled up, you do a production, you got an enzyme, and you don't have the business, how to really pitch your product, how to market your product, how to pack your product, how to address the problem of the buyer. And if you don't have the business case out of it, many good technologies, scaled up technologies have failed miserably just because they don't have the business plan in first place. So I just want everybody to really look into a business plan first and then start working towards the problem. Can we go to the next slide, please? So all young scientists learn biology. If you are a biologist, try to really understand or collaborate with engineering, economics simultaneously. None alone is sufficient for industrial success. Work on the plant floor. See to it that if you can, working with an industry, see to it that you go to the plant and observe plant how it actually operates, because that will give you some sort of a really good insight how the industry operates at their level. Imra's failure, like steady contamination, batch failures, scale up surprises, these are very important. Learn that one. If there is some fast contamination, you start working from a basis at that point. And once you understand these are the problems which might occur at a scale up level, and if you address at this moment, you are really going to be successful. Read patents, not only papers. Something very important. Patents, will you please go to the previous slide? Patents reveal what industry truly values and which commercial problems remain commercially unresolved. So read patents and build multidisciplinary thinking. That is also very important. So please try to have some sort of an good collaborations with the other laboratories which are not working in biology. So it will really give some sort of a different insight for you people. Can we go to the next slide, please? So enzyme factory of 2020 or 35 will be not the enzyme factories today. AI designs the enzyme. It will be only a few days job to really get right type of enzyme. Digital twins optimize the fermentation. Robots run the screening. Waste become the feedstock. Manufacturing is continuous. Biology behaves like a software. So the question is, will India build these factories or import them? That question we have to address. Can we go to the next slide? So industry academia gap. Academia looks for the publication, industry looks for the robustness. Academia looks for the novelty, industry pays attention to the cost per kg. Lab scale success, regulatory compliances. Isolated specialization, they are looking for multidisciplinary coming together, making a product and selling in the market. So these are the gaps in the industry and academia. And when any industry is sponsoring a project to academia, they have to really tell their pain points to in academia, and academia also has to respect what is the actual pain point of the industry. The gap can be really seen very properly. Can we go to the next slide? So shared pilot scale facilities, industry sponsored translational research, scientists exposed to manufacturing plants, startup friendly scale up ecosystem, faster regulatory mentoring, and biotech AI engineering integration. So these all ecosystems which have been created, it should be very neatly connected, and that BIRAC is doing really reasonable job in that area. So this is something which is very important. So future success will come from ecosystem, not isolated laboratories. So that is something what we have to look into. Next slide, please. Thank you.
Thank you so much, sir. I think your talk really provided valuable perspectives on industrial scalability, commercialization pathways, also the detailed process optimization and market-driven innovation in enzyme manufacturing, which will be quite helpful to our startups and industries who have been listening us. So, we just due to time constraints, we'll just take few questions. Rest the participants will get a feedback form post the webinar, where we'll be happy to answer those. As of now, just very quick questions we'll take up. First question, sir, is from Nikhil from Lemoniska Bio. This is for Prashant, sir. So the question is, I'll just read out the questions. How do you see AI being leveraged in tackling the valley of death problem so that more sustainable process can go from lab scale to industry scale? Sir, you're on mute, I think.
Yeah. Yeah. So thank you so much. Very interesting question. And what is interesting is this question was asked, but almost answered by Dr. Raghavendra Gaikavari in his last few slides. But I would just go ahead and talk about it little later in a more concrete way. Whenever we talk about use of artificial intelligence and when we talk about the process steps that are involved in development of the technology, we realize that there are various places such as strain improvement, enzyme engineering, the predicted mutations that could be beneficial. These are the places where artificial intelligence can act very well. When we talk about the pathway modification, artificial intelligence can help us very well. The reason I'm saying that is, for example, if at all I want to block the production of certain metabolites so that the entire metabolic flux could be diverted towards, for example, isobutanol production, and if I'm going to inhibit production of some of the organic acids, then which enzyme should be inhibited? For that, what primer I could use, and what could be the leader RNA sequence, and so on and so forth. I can actually gain those kind of insights using artificial intelligence. Secondly, whenever we are carrying out scale up studies, at that time, the artificial intelligence can help us in scale up studies and predicting the data, whatever data we are getting at lower scale, how exactly the behavior of the microbes or the enzymes is going to be on a larger scale. That can be done better way with the artificial intelligence. There are several such tools which are already available, and with the help of such tools from the trial or data that we are generating at 100-liter fermentation, we can directly go 100 folds higher fermentation processes as well. Thirdly, whenever so improved scalability is another point where artificial intelligence can help us. The techno-economic viability assessment can also be done in much better way with the help of artificial intelligence. Many of the parameters that we may not consider, AI tools are able to pinpoint those parameters and they are taken into consideration. So that is again another important parameter. In addition to that, whenever we are carrying out some of the studies with, say, multi-parameter optimization or using the dynamic process parameter optimization, there are various artificial intelligence tools that can be used. And most importantly, as Dr. Gaikavari pointed out, the digital twins for the scale up can be used for the better regulatory requirement identification as well. So there are several ways in which we can use various range of artificial tools for strain improvement, for techno-economic viability assessment, for regulatory requirements, and for the scalability studies. And that is how, with the help of that data, we can actually take our technologies very effectively in a way that industry desires beyond TRL 5 to TRL 6 or 7.
Thank you, sir. I think this has answered. Uh, I think the applicant is satisfied who is writing to us. Thank you so much, sir. So one more. This question is for Raghavendra sir. Sir, this is the question is from IIT Kharagpur. He's asking Dr. Narula. He's asking that I'll just merge the questions actually. So, how important is downstream processing in determining the final cost of industrial enzymes? And what exactly are the emerging market opportunities do you foresee for enzyme-based solutions globally and in India? Sir, I think Raghavendra, you're on mute, I think.
Yeah. Okay.
Yeah, thank you.
So actually downstream processing is very important. The moment fermentation is over, once you are having a broth with having an enzyme or the biomass, then if the enzyme is intracellular, then how economically you can really rupture your cells. And while rupturing your cells, how much enzyme is not going to get denatured, because the loss during the downstream processing itself is near about 30 to 40% in some of the cases. And if the loss is around 32% or 40% of that, then your cost of production is anyways going to get doubled up by simply losses during the downstream processing. So it is not only related with the financial implication, but the loss of the activity during downstream processing has to be really addressed. So downstream processing should be very properly studied with minimum shear rates, with minimum energy inputs. How we can really work it out with some enzyme downstream processing has to be worked out. Part number one. Part number two, like industrial enzymes. Now, as I mentioned you earlier, that 20 years back, making enzyme, purifying enzyme, or semi-purified enzyme, make formulation and give it to detergent industry or for tanneries or for any other industry is not the solution today. Basically, now they want an end-to-end solution. For example, if somebody is going with some sort of an enzyme cocktail for the particular application, then you should have even a pilot plant which will show them how that cost will be benefited eventually in the given process. So it should be a complete technology package along with that enzyme as one of the component of that, and then only it is going to be economically feasible. So that aspect has to be really looked into.
Thank you, sir. So we'll just take one final question for the day. This question is open, to both of you, sir. So the question is, how important are metagenomics and microbial biodiversity in discovering next generation enzymes? And what exactly strategies are being explored to improve enzyme stability under harsh industrial conditions? So, I can start, I think.
Shall I take, or maybe I will start and then Dr.
Yes,
Gaikwari
yes.
can improve on it. That way
Right, sir.
it will give a better perspective. So it is very important even in today's scenario. If you look at the NCBI database, you will see that there are a lot of metagenome sequences which are available. In addition to that, there are several genome sequences which are available. And what we have done very successfully for our industry in recent time as well is when they were looking at enzymes which are thermostable enzyme. So instead of isolating organism from some extreme environment, we directly went for the screening of the enzymes or sequences that were available in the database, and with the help of AI tools we identified the thermostable and highly catalytic enzyme, and then that enzyme we got synthesized, and then while synthesizing we made some codon optimization, some beneficial mutations that improved even efficiency further. So with that, then when we were expressing it in the host, again we used some tools to identify what exactly were the suitable host in which we could express it, and it could be secreted out extracellularly, and it helped. So what I'm trying to say here is this kind of data is extremely useful. Now you can use AI tools to actually synthesize the genes, but we have seen that more often than none, such synthesized genes have not really worked out when we express them in a bacterial host or yeast host. So what it tells us, at least at this particular time point, is AI-based tools are helpful in identifying the useful sequences from the existing sequences available in database, improving those sequences further and expressing them in a better way. Of course, scale up and other studies, techno-economic viability assessment also can be done. So as of now, I believe metagenomic libraries and such sequence availability in the database is of utmost importance, and there is no substitute.
Thank you, sir. Thank you, sir. Raghavendra, sir, you want to add to this?
Yeah, so this is something which is very important, the metagenomic data. So for example, if suppose you are working on one enzyme and you want to improve upon one domain aspect of that enzyme. For example, if you want to really go for some sort of a temperature improvement or temperature stability or a pH improvement or a pH stability, or you want that enzyme to really work in a range of pH which is comparatively slightly more alkaline than the normal pH ranges. So based on that, you can really get a complete database which is having a slightly higher alkaline stable enzymes, get that domain information through the AI tools, then develop an algorithm for that matter for that domain characters, and whatever algorithm is going to really get produced, based on that one you can really define the mutations which can happen into your own enzyme, which is a very novel and very important enzyme, but you want slightly alkaline condition for a better productivity or for some other downstream processing advantages. So that mutations can be done and that enzyme can be used. So basically getting a data, making a domain based information database first, introduce a machine learning for that matter, develop an algorithm for that matter, and get which type of mutations can really help you out in that. And that is how actually the basic data is very helpful, and that can be really used for this type of applications. Yes.
Thank you, sir. Thank you. I think it has been answered very beautifully from both the aspects. Thank you so much. So I'd like to inform the participants, due to time constraints, we have to close this, but we'll be happy to take all the questions, which you will be getting in your feedback form post this webinar. I've also put up our email IDs from BIRAC, for BIRAC officials. You can connect to us and reach out to us if you have any further queries. And people who are asking for the upcoming calls from BIRAC or biomanufacturing enablers or different verticals, please visit the BIRAC website and DBT websites regularly for quick updates. So that will be all. Thank you, sir. Thank you. I really like to thank our eminent experts, Dr. Prashant sir and Dr. Raghavendra sir, who have been instrumental in shaping research, industry, and policy in this space. So we sincerely appreciate your time, expertise, and a valuable contribution to this webinar under Bio3 policy. Thank you so much, sir. And also like to thank our DBT and BIRAC officials and all the participants who have been patiently listening to us from industry, academia, and SMEs. Thank you so much. And
Thank you.
thank you, sir, and request the participants to visit the BIRAC website for upcoming set of webinars, which will be scheduled in the second week of June. So that will be all from our end. Thank you so much. Thank you, sir.
Okay.
Thank you so much. Yeah, we are closing this webinar now. Thank
Yes.
you. Thank you.
Thank you so much.
participants.
It's been privilege for this.
Thank you, sir. Thank you.